Hi everyone, thanks for coming to the talk. So I'm Alice. I work as a PhD student at Derby Uni, looking at the role microbes and microbial communities have in the carbon dynamics of bogs and how when we restore bogs, we alter the conditions and what effect it'll have on these communities. So I've sort of come here with the assumption that people might not have the background on peat bogs or microbes. So we'll start going through a bit of that to give you a bit of context, and then we'll go into the research that myself and the wider research group are doing. So, first of all, what are peatlands? I've got a definition. So peatlands, also known as mires, are ecosystems characterized by deep accumulation of partly decomposed organic matter, or peat, resulting from carbon sequestration in plants, biomass that exceeds loss through decomposition and hydrological flow. So they cover about 3 to 4% of the Earth's surface but contain 30% of terrestrial carbon. So, peatlands hold the carbon and the stories of our past and are the key to our livable future. I quite like that quote. Um, I'm not going to go into that too much, but I'm sure you've all heard of bog bodies and the preserving nature of peatlands. So yeah, they're really intertwined with our cultural history as well as the future that we have through their climate regulation. So this is the distribution of peatlands globally. So it's predominantly in the northern zones where it's cooler and there's higher rainfall, but we've also got some sort of tropical peatlands. It looks a lot, but remember it's 3 to 5%. I think the map might suggest it looks a bit more than it is. It's only 3 to 5% of the Earth's surface. And you can see in the UK, we have a lot in Scotland, Wales, and then sort of the northern, down the Pennines, the spine of the UK, and then Northern Ireland. So what makes a bog a bog? Its hydrology is one of its key, its hydrology is one of its sort of key features. So what actually stops a bog decomposing and allowing it to accumulate that organic matter is the fact that it's waterlogged. So a lot of decomposition happens when oxygen is in a system and aerobic decomposition occurs. So if you think of a woodland, when a tree falls down and starts to decompose, that will happen when oxygen is present. In a bog, there isn't because the water table is at the surface level, as you can see in the image on the right. That's sort of how a healthy bog should look. The water is just at that top layer. So yeah, it creates anoxic conditions, which means the absence of oxygen. So it's saturated and stable. So the stability is a really key feature. So it has to be high, it has to be consistent. A lot in ecology and microbiology, a lot of conditions can survive harsh environments, but they like stability. So for your specialist species, they're not very good when things change all the time. So that those conditions are basically what create a primary productivity exceeding decomposition, which leads to accumulation. To sort of put that into context, a bog can accumulate a millimeter a year, and some of the deepest bogs in the UK are 10 meters deep. So it's the underworld, really. Like at the surface, it's a bit unassuming, but it's actually what's happening below that makes it really interesting. So in the UK, we have two primary types of peatlands. We have the bogs and we have the fens. They're both types of peat, which is where it's that accumulation. In other parts of the world, you've got swamps. So they're the three main categories: bogs, fens, swamps. So a bog is fed primarily or 100% through rainfall, so precipitation. So it's acidic and it's got low nutrients. Those two characteristics are what makes it sort of distinctly unique because the species are all adapted to these harsh conditions. And within the category of bog, you've got lowland raised bogs, blanket bogs, and quaking bogs. So the image on the left is a blanket bog. It gets its name because it sort of envelops a landscape, a lot of the uplands. So if you think of the Peak District, Lake District, or North Pennines, that's all blanket bog. It sort of spreads across the hills like a blanket. And then we've got quaking bog, the middle one, which is where vegetation forms over a body of water. So you bounce on it and it quakes. And that's a really rare type of bog. And then we've got lowland raised bog on the right. And that's formed in like domes. In the UK, we've lost about, I think, 95% of our lowland raised bogs. So historically, with peat cutting, so people used to use peat as a form of fuel. So they'd cut logs, and the lowland raised bogs were often the ones that would get cut. So we've lost most of them. And the ones that we do have, so like Hatfield and Thorn, which is the largest one in England, it's very shallow now, whereas it would have historically been meters and meters deep. It's now 50 centimeters or so. But for the rest of the talk and where my research is based is blanket bugs, so the one on the left. So just to give a bit of oversight of the fens as well, my work isn't on fens, but I'll give a bit of a description of them anyway. So what makes a fen different to a bog is where it gets its water. So it's got precipitation, but it's also got groundwater, and groundwater brings a lot of minerals with it. So it's more nutrient rich. And it's got a neutral pH, so the conditions are less stressful or less stressed. So you get a completely different mix of ecology. And then you've got rich, which is calcareous, and poor, which is acidic. So the one with the sphagnum mosses in the middle is a poor acid fen, and then your left is the calcareous. So what makes a bog unique? Partly it's the ecology. So as said, the conditions are very stressful. So you get a lot of really unique species that have adapted to survive in these challenging conditions. One of the most iconic species is the sundew, which is the third in at the top. And that's a carnivorous plant. So you can see its next prey, that little fly in the middle. So it gets its nutrients basically from consuming insects, which is really cool. And then probably the most iconic bog species is the sphagnum moss. So that is our ecosystem engineer keystone species of a bog. And basically, what makes a bog is predominantly sphagnum. It regulates a lot of the conditions. But then you've got other, birds. So the curlew, the golden plover, the large heath moth or butterfly. I think it's a butterfly, and cotton grass. So these are the sort of indicator species of a bog. And part of the reason why there is so much sort of investment and work in bogs is this rare ecology and biodiversity to help tackle the biodiversity crisis. We have sort of looking at the endangered species, and a lot of these are red list species. So in terms of why peatlands are important, this is a host of ecosystem services provided by healthy bogs. So you see there's a real mix of sort of cultural services. And this is something that the peatland sector is really good at. So a lot of artists and writers are involved in peatland work because of this link to culture and folklore especially. But in terms of things that we'd all benefit from. Climate regulation is a massive one. Water, so basically all our drinking water, I think 70% of our drinking water goes through peat bogs, upland catchments, and it sort of cleans and regulates the quality of water as well as the rate. So a lot of natural flood management is from healthy peat bogs. And then, of course, like the biodiversity, and then the provisioning. So fuel, that's where the peat cutting comes from. But I think we'll try to move away from that. In England, it's banned, but it's still going on in Ireland. So yeah. Um, so bogs gone over why they're important, but 80% of them are degraded. So they're in a pretty bad state. All these images are examples of ways in which the bogs of England and the wider UK are degraded. So top left is an example of peat cutting, where they cut out the logs. We've got drainage where channels are dug, so grips are dug across moors, and the water basically runs off like a motorway. And then we've got citrus spruce plantation. Back in the 80s, there was a lot of legislation and policy to make these landscapes productive economically. Historically, they've been deemed as unproductive. Um, we didn't really value the intrinsic value, or we didn't see the intrinsic value of the ecosystem. So there was widespread tree planting. And to be honest, we all need paper, so it has to come from somewhere. But yeah, there has been historic legislation which has, galvanized a lot of the damage. And then you've got natural forms of degradation, so gully systems, which is where the peat is scoured out by the flow of water, and then wildfire, which is becoming a much more pressing issue. So a lot of our bogs are actually at risk of huge wildfires, especially as our droughts are becoming more prolonged. The natural preventions, which would be their wetness, are going, and there's a lot of sort of above ground biomass, which makes them huge risk. Um, yes. So one way we're trying to tackle that is through restoration. So there is a big push from both the public sector, so government, and the private sector, water companies, private carbon finance, to restore the bogs because we're realizing all these ecosystem services that we get. So these are a few examples of ways in which they're restored. Plug planting, in reintroducing the ecosystem engineer, the sphagnum moss, where areas have been bare. So bare peat is a big problem because the peat literally blows away in the wind. Contour bunding, which is that top middle, and that's to sort of stop the water leaving. So if you think of overland flood, it's a structure to stop that and to keep it the moisture on the bog. And then reprofiling. Yeah, that one is peat dams, and then we've got brash spreading. So brash spreading is another form of erosion control. So there's different broad categories of restoration. So erosion control is to literally stop the carbon, being lost from site. Hydrological is to restore the hydrological characteristics, that high water table, and then sort of ecological vegetation introduction. So that's peatland, a bit of a whistle stop tour. And then I'll give you a bit of an introduction to microbes. So basically, got a definition as well. A microorganism is defined as a living thing that is so small it must be viewed under a microscope. So it's all that world of, things that we can't really see that we're actually realizing have a massive impact on the world around us. So a microbiome is the collection of all the microorganisms and viruses that live in a given environment. So they can be single cellular or they can be part of a colony, and they're split into prokaryotes, eukaryotes, and viruses. So the image on the screen is the sort of broad categories of microbes. For the purpose of this presentation, I'm going to focus on bacteria, archaea, and fungi. So a little bit of info on the difference between prokaryote and eukaryote. So basically a lot of the functional aspects of microorganisms, so the things that we benefit from as people, would be from their metabolism pathways. So if you think the one that I think everyone would be aware of would be fermentation, how we get beer, how we get kimchi, but then photosynthesis, anaerobic respiration, it's basically their means of obtaining energy. So within the prokaryotes, they're simple celled, no nucleus, and within that we've got bacteria and archaea. Eukaryotes are complex cells. They've got different energy production pathways. Within that, we are a form, and then as animals, and then fungi as well. So for peatland, for this presentation, I'm gonna discuss two of the sort of main microbiomes, so the above ground and the below ground. If you think, as I said before, peatland is sort of the underworld, and a lot of it we cannot see. Within there, imagine the amount of microbes and life that it is harboring below. So the microbiome of peat. The interesting thing about peat from a microbial perspective is the amount of different environments within a peat core. So if you think of microbes, they like different things. Some of them can live without oxygen, some can't. Their pH is a massive influence, temperature is a massive influence. So there's actually a stratification of communities based off these different environmental variables. So we broadly split it into acthelm, catatelm, and mesothelm. So the acthelm is the oxic section where oxygen is in the system. That's where active decomposition is occurring. You get a lot of your saprotrophs, sapropores, I think all your decomposers, which are bacteria and fungi, and they need the oxygen. And then your mesothelm is a zone of fluctuation where it's, half the year it might be underwater, half the year it might be in oxygen. So that'll be sort of bigger depending on drought, how long prolonged droughts are, and it's all to do with rainfall, basically. And then your catatelm basically is in constantly anoxic. It's constantly waterlogged. And that's where you get a lot of your recalcified carbon, which is this, ancient carbon that basically hasn't seen the light of day or oxygen for a very long time. And you get all your anaerobic respiration occurring there. So a lot of your methanogens, and that's where methanogenesis, which we'll go into in a bit, occurs. So, as I said, healthy bogs, high and stable. So you don't want a big mesothelm. You want it, at 20 centimeters most from surface. Um, so all these microbes are basically what determine the carbon dynamics of a bog. So the one that people would probably be most aware of is carbon dioxide as a greenhouse gas. But then for peatlands, methane is a really big one. And it's a really big consideration when you're restoring a bog. So carbon dioxide is the byproduct of decomposition. Um, but also the biggest carbon export from bog is dissolved organic carbon, which downstream is then released as CO2, but it leaves the system in water as an aquatic flux. And then methane is released through anaerobic respiration in the catatelm, where microbes, methanogens, break down carbon for their energy production. So it's their form of metabolism, their metabolic pathway. And that's the thing we have to be careful of because methane is a much more potent greenhouse gas than CO2. So in wet bogs, you've got a lot of methane coming out. But what should happen in a healthy bog is you've got a layer of methanotrophs who actually oxidize methane, and that should all live in the aquatel. So it balances 16 to 94% of the methane should be stopped at that surface level at the aquatel. So it's making sure that balance is there. When it's out of balance, then it's when all the methane gets released. So yeah, as I said, degraded bogs, these natural balances that occur are out of whack, so the emissions are a lot higher. So the above ground microbiome is sort of the vegetation layer or the microbes that live in the oxic layer. For this talk, I'm going to go into the sphagnum-associated microbiome. So as said back when I was explaining peatlands, sphagnum is sort of considered the ecosystem engineer of peat bogs. It is the keystone species, and a lot of the functionality comes from this plant. And actually, they do a lot of the methane oxidation that is being released. It'll get stopped by sphagnum, and they've got a really interesting microbiome that provides a lot of functions for the plant and for the wider ecosystem. I'm not going to go too much into it because I could probably speak all day about it. But yeah, in terms of carbon, it is this sort of methanotropic, sphagnum-associated methanotropes. So we call it a biofiltering property. So sphagnum, um, it's quite a it's a bryophyte. Bryophytes are 350 million years old, the oldest terrestrial phynological plant group. So they have survived through crazy climatic variation. They've done all the ice ages. It's managed to adapt and survive. It's like, if we're looking at our future climate scenarios, this plant has probably been through them all. And it's basically developed and evolved to have some really crazy defense systems. And in part of this is the microbiome. So one of the things with sphagnum is it gets its CO2 from the atmosphere through moisture diffusion. So it's limited when it's either too dry or too wet. This diffusion of CO2 is basically inhibited. So in theory, it shouldn't be able to photosynthesize, but it's got a secondary source of CO2, which is provided by the methanotropes that live within plant structure. So it's got a symbiotic relationship with microbes that live within the plant that can provide it as a secondary carbon source, which is just incredible. Yes, it's this biofiltering property. 16 to 95% of methane will be stopped, so the methane produced by methanogenesis, but the plant gets a lot of benefit from it as well. So now on to our research. We're basically looking at how the above ground and below ground microbiome change on a restoration site. So given the fact that 80% of the UK or English peatlands are in a degraded form, or a degraded state, our research base for this area of work tends to be on healthy, near natural peatlands. It could be completely different on a degraded bog. So we have a before-after control intervention study site that I'll introduce you to in the next slide, and looking at how sphagnum-associated microbiome would vary and how hydrological interventions will alter the microbiome of peat and what effect this might be having on carbon. So that's the title of the research: the influence of peatland restoration practices on soil and sphagnum-associated microbiome and the microbial processes related to positive restoration outcomes across an upland biotic bog. So this is all part of More Green Investment, which is our study site. So we have 4 different mini catchments on this site that are undergoing different restoration techniques so that we can see they basically have climatically the same. A lot of they have the same amount of precipitation at the same elevation, and they're so it's so high up that their water flow does not interact, so that we can basically see how different they're all responding, basically. And we have 18 months of baseline data before the restoration interventions. So this has been done with industry partnerships. So Moors for the Future delivered all the restoration across site, and they have a science and monitoring team who've been doing a lot of the monitoring. And then Derby University have been involved. So Professor David Elliott and Kirsten Lees and Dr. Alessia, we've all been working on it. And then Severn Trent, the water, you've funded a lot of it, and then the Environment Agency as well. So these are the things that we're monitoring on site. So we can see sort of the interactions of the different things. So it's quite unique in the amount of data that's being collected, which is really cool to be part of. This is basically the monitoring protocol that we have at the moment. So yeah, you can just scan your eyes over it. I'm not going to read the table. And yeah, so microbes and carbon. So the images on the screen are just some examples. So we've got a Lycor gas analyzer, which measures trace gas, so that's CO2 and methane. And then we've got some water quality work that we're doing with Loughborough University. Have I missed a slide now? So the first objective is, or the first sort of wider theme of the research, because there's objectives within it anyway. So this is looking at the below ground carbon and the microbiome. So basically how raising the water table will alter the microbial composition and how that'll change the carbon balance and whether it is actually due to microbes. So with this sort of thing, you have to factor in what else might be changing the carbon dynamics. So one thing we're looking at is the phenolic compounds, pH, the actual carbon content of the peat, as well as all the microbes. So yeah, we've taken cores across the site, split them into 10 centimeter chunks, extracted the DNA, and then sequencing it, which we're using 16S sequencing, and that'll provide community composition for the bacteria in archaea. And we can see pre-restoration, post-restoration, and then in 2026 as well. And then how that links to the changes in carbon that we're observing as well. So the carbon measurements, so the one on the left is particulate organic carbon, and you can see that there's a huge difference. Just visually, you can see that there's a massive difference, and that has been collected from artificial pools, which are what you basically get when you do either bunding or damming. You will get a small body of water forming behind that intervention. We've taken samples from dip wells, which are basically go into the peat core. They are to measure water table depth, but it's quite a good way of getting peat pore water as well. And then we're measuring the outlet streams of each of the catchments. And then we're going to be looking at dissolved organic carbon. So that's the stuff that, I don't know if you get to the Peak District much, but the water sort of looks a bit like treacle sometimes. So that's dissolved organic carbon. And that's a big problem because when actually trying to remove the colour, discoloration, they have to use chemicals which I believe are carcinogenic in the wrong quantities. Like it's obviously fine for us to drink, but we obviously, if we can have a way of removing it before it enters the system, that would be preferred. So we are actually seeing massive changes in the methane. So the one on the left is the before, and then the one on the right is quite hard to read from that distance. The one on the right is the after. So you can see there's been massive changes in the microbes, and this we can see that it is aligning with where the water table has shifted from to. And then it actually has been reflected quite a lot in the methane fluxes. So that really big spike, that's in the summertime. So a lot of microbial activity is influenced by temperature. So you will get spikes in sort of summer because it's warmer. And so we're seeing quite nice seasonality in the monitoring. but there is a big spike, and we're hoping that eventually it'll sort of ease off a bit once the methanotrophic communities, which oxidize methane, return. So it's in a sort of um, during restoration, it's trying to find a new equilibrium. So it is disturbed still, and it will take time to recover. And then CO2 has been really interesting because we're seeing quite a lot of spikes. And there's a chance that that could be because there is a lot of fungal activity at the surface. So I've been reading into this quite a bit. And when there is a lot of fluctuation, as I said, bogs like it's stable. When there's fluctuation, it favors fungi over bacteria. So we want the bacteria because they're the ones who oxidize methane. But when it's unstable, you'll get an influx of fungi, which they call fungal invasions, which I quite like. But yeah, that could be linked to that, and we're going to be looking into that a bit more. So the sphagnum associated microbiome is sort of the next objective of the project, which is starting around now. So I don't really have anything to tell you about it. But it's basically looking at the biofiltering property of sphagnum and the role water table plays in establishment of sphagnum-associated methanotrophs and the functional implication on both the host plant and the wider ecosystem. So we've got a study that we're setting up at the moment. So it should be going out in situ on Monday. Fingers crossed the weather plays ball. Because we can't plant them when it's frozen. So I'm hoping a bit of a warmer temperature. Yeah, we're going to be looking at how the microbiome of sphagnum establishes on a restoration site. So this is the study design. So around automated dip wells, we're going to plant three species of Sphagnum cuspidatum, Sphagnum capillifolium, and Sphagnum papillosum. So they're three different functional categories of sphagnum. So one is a lawn, one is a hummock, and one is a hollow species. I won't go into all that now, but basically there's different microclimates across a bog. So we wanted to see if there is actually a difference in these different species because we have observed data that they provide different ecosystem services. So a lot of that could be due to their different microbiomes. Anyway, so each plug we're going to be monitoring with hyperspectral imagery. So we're doing that with some partners that we're working with Prioptics. So we can see we've got a ton of health metrics that we can look at. And we're looking at size through segmentation and seeing how fast they grow. As well as things like moisture content, water holding capacity, and then all the microbial data on top of that using quantitative qPCR, so real-time qPCR, which is basically a microbial technique where if you've got like a functional gene, in this instance, it'll be methanotrophs, so methane oxidizers. We can tell the machine we want to look at this, and then it'll basically tell us abundance. So you can track the abundance of this species and if it's changing and if it's different across the different sites and across the different species. So this is a little bit about concept development. So we've been trialing DNA extractions because it's a bit more complicated with a plant because you've got a lot of plant material, chlorophyll. So it's a way of trying to get the microbes and, from a living plant, basically. It's a bit more complicated than peat. And then that image, top left, is the hyperspectral device that we've been trialing on site. And these are some examples of mesocosms that I've been playing around with in the greenhouse to sort of establish the protocol before the plugs go in on Monday. Cool. So basically, yeah, watch this space.